Hello everyone. In this video, we're going to see how plants fix CO2, and eventually they use that to make glucose. So this is the synthesis part of photosynthesis that we're going to be talking about. Let me move myself just a sec. Okay, here. All right. So the overall reaction is shown here, and it makes the whole point of why we have photosynthesis. This is CO2, and it's going to become glucose, 6 carbons, all by themselves, single units to one um, 6 unit, 6 carbon unit. That's synthesis, right? Stitching them back one to one to one next to each other. It doesn't exactly happen like this in a sequence. We'll see how it happens, but the net reaction, the overall net reaction, is the generation of glucose from single carbon units. And for that, as for every photosynthesis, as for every biosynthetic process, you need two things. You need energy. Okay, you need the little building blocks, which is the CO2 in this case, and two more things. So you need energy, currency in the form of ATP. 18 of those will come into place to make one um, uh, glucose molecule, and a lot of reducing power. NADPH, 12 of those, which of course you got from photosynthesis, from the photo part of photosynthesis. So this reaction literally summarizes the general principles of any biosynthesis that our cells do, which is really you have little building blocks, um, currency and energy, currency, and electrons, reducing power. Let's see how this happens. It happens in a sequence of reactions. As I said, it's not just you don't just add one carbon next to each other. It's a rather complex series of reactions, and it goes into a cycle. And the person who discovered them was Calvin in the 40s and 50s, got the Nobel Prize for it. There he is at his lab at UC Berkeley. So the overall reaction, again, it's not complete. It's a very simplified scheme. We're going to see how everything happens, but you start with a 5 carbon unit, um, ribulose 1,5 bisphosphate, as it turns out. So you have 5 carbons plus 1 carbon from CO2, and gives you a 3 carbon unit, okay, 3-phosphoglycerate, and that gets converted to glyceraldehyde 3-phosphate, which we have already seen those two things, 3-PG and GAP. We have seen them in glycolysis. They're glycolytic intermediates. And of course, we have input for this to happen. You have input of ATP plus NADPH. So as I told you, to get to carbohydrates, you don't really go one by one carbon plus 1 carbon plus 1 carbon until you get to 6 carbons. Okay, you have 5 plus 1 gives you 2 times 3, and so that's a net gain of 1 carbon. And then the goal is to come back, so you can keep doing this. You have to regenerate this 5 carbon unit, okay? And that's actually a whole series of reactions, how that happens. And it's necessary in order to close the cycle, so that you overall, you keep doing it, you keep turning the cycle, let's say 6 times, then you're going to have the generation of a net 1 6 carbon unit, like in the form of glucose, right? So it's quite complex, but we'll see how it happens. So the first reaction, the one that actually fixes carbon, is catalyzed by an enzyme called Rubisco. And so it takes a 5 carbon unit shown here, ribulose 1,5 bisphosphate. So it's a 5 carbon unit phosphorylated on both ends. Uh, and to that, it adds one CO2. Okay. So we have a carboxylation of a 5 carbon unit, gives you transiently a 6 carbon unit, which, as I said, very quickly once it's made, then it becomes it gets split into two 3 carbon units, glycerol 3-phosphate, which we have seen already. Excuse me, from glycolysis. It's now you don't see any ATP or NADPH yet here, okay? Because then when you take glycerol 3-phosphate and you take it up to make the glucose that you need, reversing some reactions in glycolysis, to actually literally make gluconeogenesis, this is when you actually need it. So in the first reaction by Rubisco, you don't need ATP and NADPH. Now, Rubisco is the most abundant protein on Earth. Of course, it's in every green leaf that you see. Um, it's, for example, okay, it's rather large. It's about half a megadalton. But it's also a crappy enzyme. It's really slow. Okay, it's got a catalytic rate of 3 per second. That's really slow when it comes down to enzymes. I mean, we've seen many faster enzymes. And for this reason, there has been an enormous effort to improve Rubisco. And if you had a penny, as the saying goes, if you had a penny for every attempt to make Rubisco a better enzyme, you would be a millionaire now. But the obvious implication here, if you can make it, faster, the faster the plants will be able to fix carbon, the more product you're going to get, sugars and so on and so forth. So it has an obvious application. It's also extremely highly regulated, okay? And we'll see how that happens. It needs magnesium for its activity too. And we'll see how that happens too. All right, what is it? I've already told you it's a carboxylase. You make carbon-carbon bonds. I mean, we've seen several of those already. The mechanism is a little different in this case. So how does it happen? Well, it happens. It still has to satisfy the enzyme has to solve a few problems that we have seen before. What's the species of CO2, and well, it's CO2. It's up in the air. Okay, then you have to activate it. It gets in, and then it has to be activated. It's a pure electrophile. So in this case, it gets activated by metal-ion coordination. And then how do you stabilize? And for this activation, you need magnesium, as it turns out. And so activating the CO2, you need magnesium for that. Otherwise, it doesn't work, the enzyme. And the last problem is to stabilize the carbanion. So how does it get stabilized? We've seen the mechanism before too. It's a ketoenol tautomerization. So this is the substrate, RuBP, rubulose 1,5 bisphosphate, and it looks like this. So exactly as you see here, you're going to make the intermediate. This is where the carbanion will be formed. So the more it can bounce up and down or left and right in these different tautomers, the more you stabilize it, right? So because it has this option, so it gets stabilized. You can go back and forth. And now in this conformation right here, that's the carbanion and it can attack CO2. So that's how you stabilize it, right? So problem solved, right? So you stabilize the carbanion, you activate the CO2 with magnesium, as we said. So the problems are solved, and you can proceed. The way it proceeds is shown here. So that's the carbanion. It will attack the CO2, and there's your CO2, and then almost immediately, okay, it gets split into 2-3 carbon units in the scheme shown here. Okay, so this bond now, so when you the keto group that was formed here, when the carbanion attack the CO2, that one, okay, will keep, it will be hydrolyzed essentially, this carbon-carbon bond here, and that keto group will play a role. Okay, so first you have hydrolysis. You have the attack of water on that keto group. It will open up, and then when it closes back up to make the keto group, then this carbon-carbon bond will go away. And you generate your first 3-phosphoglycerate, and then you have a carbanion of a 3-phosphoglycerate. All it has to do is pick up a proton, and it becomes another 3-phosphoglycerate. So the bottom line, you got a 5-carbon unit. We saw how the carbanion is formed and stabilized to attack a CO2 molecule, and you carboxylate your 5-carbon unit. You get a 6-carbon unit. This is unstable, it's very transient. So you have an attack of water again. So you hydroxylate first, and then you break the carbon-carbon bond. Okay. And once you break it, you have 3-phosphoglycerate, 2 molecules of 3-phosphoglycerate forming. How is it regulated? The enzyme is on at day. So when there is photosynthesis going on, the enzyme is on. At night, it gets turned off. So what happens in photosynthesis is in the photo part when it's um, daylight and when the sun shines, we have the electron transport chain, and which we discussed in previous videos. And this is supposed to be the membrane of the thylakoid. So at the bottom of the slide is the lumen, the inside. The top is outside, and this is when there is a lot of protons that get pumped in, get translocated into the inside from the stroma into the lumen of the thylakoid. Right. So really, the thylakoid during the day has a lot of protons inside, and turns out that the magnesium is outside, okay, in the stroma. At night, the thylakoid is somewhat leaky, so things don't stay, super tight forever. After a while, what happens is magnesium comes in and protons go out, okay, at night. Now, Rubisco, I just told you, needs magnesium. So it turns out that Rubisco is actually in the stroma, not in the thylakoid. So Rubisco is in a place where it has a lot of magnesium during the day and lower magnesium during the night. Because it needs magnesium, that's one way to turn, let's say, Rubisco off at night, okay, in the stroma. In addition to be activated, Rubisco needs to be post-translationally modified. Another CO2, a different CO2, not the one that participates as a substrate, in the carboxylation of 5-Ruby-P. But another CO2 comes on and modifies, it carbamates a lysine residue. Okay. So this CO2, not to be confused with the substrate of Rubisco that we saw previously, when we made the 2 molecules of 3-phosphoglycerate, but a different CO2, this one does not get really, used in biosynthesis to making more glucose, but it's used instead to modify Rubisco itself. So a different CO2 is modifying a lysine, and for that you also need magnesium, as it turns out, to properly coordinate the CO2 that gets added onto the lysine residue, and it is this carbamated form of Rubisco that's actually active. So there again, you need the magnesium for activation of Rubisco. And where do you find the magnesium? You find in the stroma and at night, during the day, not during the night. And it's also favored, this reaction, as it turns out, by low concentration of protons. Again, you are in the stroma. Okay. Low concentration of protons during the day. Why? Because all the protons get translocated in to the thylakoid inside in the lumen of the thylakoid during the day by photosynthesis. So you've got low and low protons and high magnesium. So that explains the activity that you see of Rubisco during the day. One more way that it's regulated. So the cell goes into extraordinary lengths to have this tight regulation. So it makes an analog, a transition state analog shown here, CA1P. So it looks very similar, okay, to the transition, like to the transient compound that you are forming there. It's got the 6 carbons. There you go, 1, 2, 3, 4, 5, 6 here. So this looks almost the same, okay, as the normal transition state compound that you form during the course of this reaction of Rubisco. So, and that goes into the active site, it binds there and inhibits the enzyme. And that is made actually at night by the plant. So during the night, I mean, the Rubisco is off in a variety of ways, as we discussed. All right, so something else that is happening when you have CO2, Rubisco will go through this reaction that we just discussed, and it will give you the 2 molecules of 3-phosphoglycerate. All right, and we have not seen the rest of the reactions, but we'll see them in the Calvin cycle. But Rubisco can also incorporate the oxygen instead of CO2. And in that case, you have what is called photorespiration. So oxygen is consumed, okay? But you have no fixing of carbon at that point. Right? So you generate these products, 2-phosphoglycolate and one molecule of 3-phosphoglycerate, which then goes on to become glycine, and from there on to serine, and then glycerate, and then it turns back into RuBP, and you can keep going like this. You have then this photorespiration cycle. So when that happens, you don't get any net carbon fixation. So which one is it? I mean, there is both oxygen in the atmosphere. Which one happens in real life? Okay, there is both oxygen and there is CO2. In fact, a little bit of both, a little bit of some of photorespiration will always happen. Okay, what you hope is to do a lot more of, if you are a farmer and you grow your plants, is you hope that your plants do the Calvin cycle as opposed to photorespiration, so they can increase the sugar production. Some cyanobacteria, the way they do it, they're very efficient, okay, and when they do it. So they bring in CO2 from the air. It becomes carbonate once it's in the cell, and then they put all the CO2 in a specialized compartment called carboxysome. It's not a membrane-bound organelle, but it's more of like an aggregate, and inside the cell, inside the cyanobacterium, called the carboxysome. And actually, this is where Rubisco is. And the carbonate is brought in, then it becomes CO2, and it finds exactly, so it says you have an infusion of Rubisco. You have a Rubisco aggregate that then gets infused with CO2 and doesn't see too much oxygen to begin with, and that makes it very efficient, actually. And you drive specifically the Calvin cycle as opposed to photorespiration. Right? It turns out if you transfer cyanobacterial carboxysome into green plants, you do increase their production, okay? And you have to have a specific pump. And where do you put it? Sorry, you put it inside the bacteria-like compartment, okay? The chloroplast that plants have. As long as you put the specific pump and you pump in the carbonate right exactly where you want it, and you make the carboxysome, then it seems to be working. So you take the green and the blue parts from bacteria, you stick it into plants, and now you can have your Rubisco infused compartment, and which turns out to be much more efficient because again, you concentrate the CO2 where Rubisco is, so that's all it sees, and you force it in a way to do the Calvin cycle, not photorespiration. Photorespiration, however, increases with temperature. So whether Rubisco is going to use CO2 or oxygen depends on temperature. The higher the temperature, the higher the photorespiration rate. Right? Now, if you're a plant and you live in a hot climate, let's say like Texas, or even worse, in more tropical environments, you do a lot of photorespiration, and that's going to be detrimental for the plant because you don't end up not fixing much carbon. So how do tropical plants, let's say cacti, do it? Well, they do something very similar to what the carboxysome does for cyanobacteria. I mean, the concept is actually the same. What they do is they compartmentalize where Rubisco is, and they put CO2, as much as CO2 as possible, right where Rubisco is. So they have these are the two layers, let's say, of the leaf. So instead of putting Rubisco right on the outside, on the outer layer, okay, which maybe there that's going to get a lot of oxygen and it's going to drive photorespiration instead of the Calvin cycle, they do other reactions that we have already seen. They take phosphenol pyruvate, okay, and we saw this in the anaplerotic reaction of the TCA cycle. You need to replenish a C4 compound, a 4-carbon unit, which is oxaloacetate, right, in the TCA cycle. And how do you do this? You take an activated 3-carbon unit like phosphenol pyruvate, and you carboxylate it. Okay, we've actually described this reaction in detail, and you get a C4 compound, okay, oxaloacetate, let's say in this case. And then, if you manage to decarboxylate it, but the way that works is decarboxylated in the next layer of cells, the bundle seed cells, as opposed to the mesophyll cells on the leaf. And this is where Rubisco is. In a sense, you infuse, again, Rubisco with CO2, and then you help it to drive, the cow to go sort of, you navigate it towards the Calvin cycle and away from photorespiration, and that makes it very efficient. And now they can produce sugars. So this is called C4 photosynthesis. Again, the idea is to put Rubisco to put Rubisco in a place where it can see mostly CO2 and not so much oxygen, right? Which is in principle is what cyanobacteria do, right? So it turns out that many, not just cacti, but agriculturally important plants like sorghum, which a lot of it is actually grown in Texas. It's used for feed, for animal feed for the most part. But in Africa, where it came from, in the Sahel region, it's actually a food staple for people. Do this kinds of photosynthesis, the C4 photosynthesis, and the future with the climate change and the increased temperatures and on the planet is really to engineer varieties, crops that can do this, so they can be more efficient and the yield can be increased. All right, so let's assume you solve all these problems. So what we got out is really 3-phosphoglycerate, which again is a glycolytic intermediate, but it has to go back. We haven't seen any glucose yet, right? So how does it go back? We've seen these reactions. This is from glycolysis. In this case, we went from left to right. Okay, glyceraldehyde 3-phosphate. It becomes 1,3-bisphosphoglycerate and then 3-phosphoglycerate. So, but here we have to go in reverse, right? And already you can kind of see where the ATP and the NADPH will come into play. But let's see it. By the way, this if you go from left to right, we have glycolysis. If you go from right to left in reverse, we have now the parts of the Calvin cycle. So how do you go? How do you reverse this? Well, we had seen phosphoglycerate kinase, okay, that it was used in glycolysis to make ATP, but it can go back if you give it a lot of ATP, from which we got a ton from photosynthesis. If you have the ATP to spend, you can reverse it. And how do you reverse the next reaction? Well, I mean, you have to give it NADPH in this case. The way we had seen in glycolysis, it was NAD plus that was converted to NADH. And in fact, to replenish the NAD plus, this is why we had fermentation. Here we go in reverse, and the source of electrons, in this case, is NADPH, which again, you got a ton from photosynthesis, so you can actually drive it backwards. So this was the redox reaction in glycolysis that we had discussed. So again, to reverse this reaction, okay, you need ATP and reducing power in the form of NADPH, which again you got from glycolysis. The rest is the upper part of glycolysis, and it goes in reverse. And it's actually super easy to go in reverse, except one reaction right here, okay, the phosphofructokinase reaction. How do you reverse that? And the way to reverse it is to just come up with a different reaction altogether. So instead of doing the reaction as a kinase shown here, which is a phosphofructokinase, you have to make another enzyme, a phosphatase in this case, which is very specific in removing the phosphate at position 1 that PFK normally puts on, all right, of fructose. So the FPP, the phosphatase, will remove this phosphate, and then you got fructose 6-phosphate, and once you got fructose 6-phosphate, you can very easily go to glucose 6-phosphate. Those are just simple isomerization reactions. As it turns out, FPP, which we will see also, we have this enzyme. Okay? When we do gluconeogenesis, the making of glucose, it involves all these steps. And so it's regulated allosterically, just like PFK is. However, whatever was an allosteric activator of PFK, such as AMP and fructose 2,6-bisphosphate, is now an inhibitor, okay, of allosteric inhibitor of FPP. So this is, I mean, the reason for this is obvious, right? So you want to, if you have these compounds here and you want to take them down to glycolysis. At the same time, you want to inhibit whatever will effectively reverse the pathway and take you back to glucose. So this is how you get glucose, but only 2 out of the 12 glyceraldehyde 3-phosphates is used to synthesize glucose. We made 2 glyceraldehyde 3-phosphates from the Rubisco reaction, and it's those 2 that, only those 2 that actually used to make glucose. The rest, why do we need them? The rest, you need them to regenerate the 5 carbon units so that Rubisco can work again. Right? I mean, it makes, yeah, let's go back. Wow, it's all the way back. Right here. I mean, so we got the 3 PGs, but and that gives you the sugars, but from one reaction. But once you do that, and you make 1 glucose, then you cannot go forward. So you need to keep making this more 3-phosphoglycerates, so you can reverse, you can generate the 5 carbon unit that you absolutely need to keep fixing carbon. So in the next slides, in the next video, we're going to see how that happens. So we really effectively have to close the circle. Okay, I went all the way back. I should have just stayed on track with the slides. And that's the part where we're going to be closing the Calvin cycle.